Welcome to today's explainer. We're taking a completely cinematic 3D journey into the microscopic world. Imagine zooming in, past layers of human tissue, down until you're face to face with a single glowing eukaryotic cell. Every living organism out there, from the tiniest microscopic bacteria to the massive blue whales in our oceans, is built from this one fundamental unit. Today, we aren't just going to look at the cell as some static blob, but as a dynamic, highly compartmentalized, and incredibly efficient microscopic factory. Okay, let's dive into this. Here is our roadmap for the journey. First, the fundamental unit. Second, boundaries and control. Third, the cellular production line. Fourth, powerhouses of the cell. And finally, structure, movement, and maintenance. All right, part one, the fundamental unit. Before we can really appreciate the cell's ultra-detailed machinery, we kind of need to know how we even found it, right? It all started when Anton von Leeuwenhoek actually observed living cells for the very first time. Then Robert Hooke gave them their iconic name, cells. That paved the way for three pioneering scientists, Schleiden, Schwann, and Virchow, to formulate the cell theory. They realized that absolutely all living things are made of these fundamental units. It was a unifying idea that changed biology forever. And the biggest takeaway from that era is perfectly summed up in this famous Latin phrase formulated by Rudolf Virchow, omnis cellula a cellula. It translates to all cells arise from pre-existing cells. If you're preparing for exams like NEET, you absolutely want to burn this into your memory. It anchors the entire concept of life's continuous chain. A cell doesn't just magically pop into existence, it is always born from another cell. Moving right along to part two, boundaries and control. Look, any efficient factory needs a really secure perimeter. First up is the plasma membrane. If you look at this in a dynamic cross-section, you'll see Singer-Nicholson's fluid mosaic model. It's a fluid phospholipid bilayer with polar, hydrophilic heads and nonpolar tails, offering selective permeability. It actively chooses what gets in and what gets kicked out. On the flip side, plant cells, fungi, and algae have an outer cell wall. It's rigid, non-living, made of cellulose or chitin, and it provides some seriously incredible mechanical support. Now, zooming right past that boundary, we hit the absolute heart of the cell, the nucleus. Discovered by Robert Brown, this is basically the CEO's office. Enclosed by a double membrane with specialized nuclear pores, it protects the genetic instructions. Inside, you've got the nucleolus working hard to synthesize ribosomal RNA, and it's all surrounded by unraveling chromatin fibers. Those fibers hold the DNA, literally the ultimate genetic blueprint needed for hereditary and every single cellular function. Next up, section 3, the cellular production line. And this brilliantly illustrates how the cell actually builds things using a highly coordinated endomembrane system. First, panning over to the rough ER, studded with ADS ribosomes, we see it synthesizing proteins. Meanwhile, the smooth ER, which has no ribosomes, handles lipids and steroids. Once those products are made, they flow right into the Golgi apparatus, discovered by Camillo Golgi. These flattened cisternae act just like a shipping center, modifying, packaging, and routing everything. Finally, the Golgi forms lysosomes. These are our acidic suicide bags, packed with hydrolytic enzymes to dissolve cellular waste and debris. But what about plant cells? Take a look at this massive number, 90%. In plants, large central sacs, called vacuoles, can take up almost the entire volume of the cell. It's kind of like a giant water balloon inside. And it's definitely not wasted space. These massive vacuoles are absolutely critical for maintaining turgor pressure, which keeps the plant standing upright, and for storing excretory products. All right, on to part 4, powerhouses of the cell. So our cellular factory has a perimeter, a control center, and a production line. But as we know, a factory simply cannot run without power. Both mitochondria and chloroplasts are double-membraned, semi-autonomous organelles containing their own 70S ribosomes and circular DNA. That is a vital detail to remember. It means they're essentially independent mini-factories operating right inside the larger cell, totally distinct from the main nucleus. Let's contrast these two incredible energy generators. First, the mitochondria. It generates ATP, the cell's main energy currency, via oxidative phosphorylation along its highly folded inner membranes, called cristae. Plant cells, on the other hand, rely on plastids, specifically chloroplasts. Picture stacked thylakoids or grana packed with glowing green chlorophyll and bathed in fluid stroma. It's perfectly engineered to drive the amazing process of photosynthesis. Bringing us to our final section, part 5, structure, movement, and maintenance. So what holds this entire 3D structure together? Well, you've got workers on the factory floor, the ADS non-membrane-bound ribosomes, discovered by George Palade, busy assembling proteins. But the actual physical scaffolding? That's the cytoskeleton. It is this dynamic, elaborate network of microfilaments, intermediate filaments, and microtubules. Not only does it maintain the cell's precise shape, but it also acts as an intricate microscopic highway system for intracellular transport. It's literally what keeps the whole factory from collapsing in on itself. Which leads us to a pretty fascinating question. We know the cell is packed with complex machinery, but how does this microscopic factory actually move through its environment? Or maybe even more importantly, how does it physically divide to create new life? Take a second and just think about the sheer logistics required for a cell to orchestrate its own division. Now, what's really interesting about this is how the cell solves these logistical problems using strict mathematical arrangements. For cell division, animal cells use the centrosome. It contains two perpendicular centrioles with a precise 9+0 microtubule organization to form the vital spindle fibers. For movement, cells use cilia and flagella, hair-like outgrowths utilizing a 9+2 axoneme arrangement. If you're an exam learner, lock this in. It's 9+0 for centrioles and 9+2 for cilia and flagella. Those are highly tested concepts for sure. Finally, we have microbodies. These are minute, single-membrane-bound vesicles, like peroxisomes. You can think of them as the ultimate detoxifying cleanup crew. They contain specialized enzymes for breaking down fatty acids and neutralizing harmful substances like hydrogen peroxide, basically keeping the entire cellular factory safe, clean, and harmonious. We've seen how absolutely incredible just one single cell is, from fluid membranes to ATP-generating cristae and those complex microtubule highways. It really is a marvel of 3D biological engineering. So I'll leave you with this. If every single cell operates as a perfectly calibrated, autonomous, microscopic factory, what does that make the complex organism it builds? I mean, you and I are made of trillions of these units, all working in absolute perfect synchronization. Keep that perspective in your mind as you continue exploring the breathtaking world of biology.
Let's jump right into this explainer and explore the absolute marvel that is the endomembrane logistics system. We're going to look at exactly how eukaryotic cells modify, package, and ship biomolecules, and honestly, they do it with a level of perfect precision that modern human technology can literally only dream of.
To kick things off, I want you to imagine a city. A bustling, sprawling city that manufactures millions of distinct chemical products every single second. We're talking vital stuff here: enzymes, hormones, membrane receptors. Now imagine this same city delivers every single one of those products to its exact target without ever losing a single package. So what's the most efficient factory on Earth that could possibly pull this off?
Well, it isn't a futuristic Amazon fulfillment center. It's a single eukaryotic cell. And this just perfectly captures the sheer, mind-blowing scale of cellular biology. We aren't looking at some AI-driven mega warehouse. This is the microscopic reality happening inside billions of your cells right this very second.
Let's start with part 1, the cellular factory floor.
Okay, so every manufacturing process needs a starting line, right? Synthesis starts right here at the rough endoplasmic reticulum, or RER. Notice how it's studded with these little structures called ribosomes. You can think of those ribosomes as our tireless factory workers. They're docked right on the outer membrane of the RER, and their whole job is to synthesize raw protein materials directly into the RER's internal chamber, which we call the lumen.
The second that newly synthesized protein cargo enters the lumen, the factory floor comes alive. A highly coordinated assembly line instantly kicks off, and it always follows 3 immediate steps: protein folding, initial modification, and quality control. Let's break down what's actually happening at each station.
During step 1, protein folding, things get surprisingly physical. You have these molecules called molecular chaperones on site. Raw polypeptide chains are basically useless floppy strings until they take the right shape. So these chaperones step in to physically assist the protein chain, bending and folding it into its functional 3D conformation. Because without that specific 3D shape, the protein simply won't work.
Now for step 2. I know a term like N-lint glycosylation sounds like a total mouthful, but it's really just the cell's way of adding some early accessories to the product. During this initial modification, short sugar chains are snapped on to very specific amino acids on the protein. It's laying down the foundational chemistry that dictates how this protein will behave later on.
But hey,
A factory is absolutely nothing without quality control, and the cell, it is ruthless about this. If a protein misfolds, it doesn't just get shoved down the line. It gets chemically tagged on the spot and sent straight for degradation. Defective products are destroyed before they can ever hit the shipping lane.
All right, moving on to part 2, the ultimate distribution center. To actually leave the factory are folded, tagged cargo buds off the ER. But it doesn't just float around randomly. It gets packed into COPII-coded transport vesicles. Think of these vesicles as microscopic delivery trucks. They drive along the cytoskeleton, which acts like a literal highway system inside the cell, until they arrive at the Golgi apparatus. The Golgi is basically the cell's master chemical processing facility. The Golgi has a very strict directional flow. Vesicles arrive and fuse at the cis face, which is the receiving dock. From there, the cargo is processed through these flattened sacs called cisternae. It moves through the medial section until it finally hits the transface. That's the shipping side, where things are fully prepared for dispatch. As cargo moves from the cis to the trans side, it undergoes some really heavy modifications. The Golgi performs enzymatic trimming and O-linked glycylation, where sugars are altered, trimmed down, or expanded to form highly specific oligosaccharide chains. And, just as importantly, it does molecular tagging. Precise chemical tags are attached to the cargo to act as literal destination labels. Let me give you a great example of this tagging. If the Golgi slaps a mannose-6-phosphate tag onto an enzyme, that acts exactly like a priority overnight shipping label. That chemical signature guarantees the enzyme will bypass everything else and be routed straight to a lysosome, which is the cell's waste processing plant. It's a foolproof barcode system made entirely out of chemistry.
Which brings us to part 3, final delivery target zones. Welcome to the Trans-Golgi network, or the TGN. This is the ultimate outbound shipping terminal. Here, the completed packages are sorted based on those destination labels we just talked about. They get packed into specialized vesicles, often coated with proteins like Clathrin. Once they're sealed up, they pinch off from the Golgi, ready for final delivery.
Depending on their labels, these Clathrin-coated delivery trucks are routed to 3 distinct target zones: constitutive secretory vesicles, regulated secretory vesicles, and lysosomes or endosomes.
Let's quickly break down these 3 targets. First, constitutive vesicles move continuously. They're constantly fusing with the plasma membrane to release structural materials and rebuild the cell wall. It's 24/7 maintenance. Second, you have regulated vesicles. These are incredibly patient. They wait right at the membrane for a specific cellular signal, like a calcium spike triggered by a nerve impulse, before they release crucial contents like insulin or neurotransmitters. And finally, the lysosomes receive hydrolytic enzymes to act as cellular security, breaking down waste and foreign invaders.
When you step back, it is genuinely awe-inspiring. The vital hormones and enzymes keeping you alive right now are the result of an incredibly advanced assembly line, a highly precise chemical lab, and a flawless logistics network. And it's all completely integrated into a space so microscopic that you need advanced technology just to see it.
It leaves you wondering, doesn't it? If a single microscopic cell can manage this level of perfect, unceasing efficiency, millions of deliveries a second without a single computer or barcode scanner, what other incredible secrets of engineering are hiding right inside our own biology? Thanks for exploring this with me today. Keep asking those big questions, and I'll catch you in the next explainer.